Amygdala, VMPFC, and threat. To think a bit more about our extinction in relation to clinical conditions like phobia or post-traumatic stress disorder, PTSD, let us first think about how Pavlovian conditioning works in the context of threat. So instead of pairing neutral stimuli with a rewarding or positively appealing US, like food, experimenters have also paired them with a potentially frightening or aversive US, like an electric shock. After a CS has become paired with such a US, it may elicit responses in rodents such as freezing behavior, meaning they may stop moving completely. In humans, we can measure their skin conductance response, SCR, which increases as we are afraid, because we would be sweating a little and increase our skin conductance for electricity. Another medial temporal lobe region, the amygdala, is important for this kind of learning. In fact, like the SCR, activity in the amygdala also shows responses to a CS that has been paired with the threat. Importantly, we also know that the amygdala receives inputs from the VMPFC. So in some experiments, mice learned that a tone, CS, was paired with a threatening electric shock, US. Now after this conditioning phase, CS alone elicited amygdala activity. Accordingly, the mice would freeze too, as if they were scared when the CS alone was presented, and amygdala activity reflected this behavior. Now then, to extinguish this response, we could present to them the CS without the US for extinction training. We know that mice can learn that. But in an experiment using optogenetics, it was found that when we use light to inhibit activity in the ventromedial prefrontal cortex, VMPFC, during this extinction phase, it took much longer for them to stop showing freezing behavior to the CS. In fact, some animals never cease to show such response to the CS under inhibitory optogenetic stimulation to VMPFC. This suggests that the VMPFC may be important for acquiring extinction memory. Specifically, such learning, unlike the acquisition of the CS-US relationship, does not happen entirely within the amygdala, but rather VMPFC sends an inhibiting signal to the amygdala as if it were telling the amygdala, we don't need to be scared anymore. Because in this context, the CS is no longer paired with the US, but the amygdala itself may never forget that the CS and US were paired at some point.